Good morning, everyone. My name is Shreya Jariwala, and I am an intern at Morphobank. And today I'll be talking to you about AI-assisted data transfer, moving data from the paleobiology database to Morphobank. And so let me begin with the table of contents and give you an idea of what I'm going to be talking about today. So I'll be giving you a slight introduction to Morphobank. I will be talking about the problems that are associated or that we're facing at Morphobank currently, an AI-powered solution that I came up with to battle these problems, kind of the mechanics of how it works in the background, future applications, and then I'll be open to questions through email. I'm so happy to be able to present, even if it's virtually. So if you guys have any questions at the end of this presentation, please feel free to reach out to me through email. So let me begin by a little description of what Morphobank actually is. And so Morphobank is a powerful online platform that is designed for collaborative research in phylogenetics. This is kind of a part of a larger movement that's known as the FAIR data movement. It's in order to make data more findable, accessible, interoperable, and reusable. Morphobank is, of course, an open access repository specifically designed for phylogenetic matrices. So this part of FAIR is just for phylogenetic matrices, which makes phylogenetic matrices findable, accessible, interoperable, and reusable. And so this is a quick overview of the functionality of Morphobank. So scientists will be able to upload their phylogenetic matrix onto Morphobank. A phylogenetic matrix usually contains taxa, character names, character states, and of course the matrix. And there are a couple of additional functionalities kind of put into Morphobank to be able to get more information for this particular matrix that you're uploading. And so we've partnered up with the Paleobiology Database and NCBI to be able to add more information regarding the taxa that you upload. So let's say your matrix only contains the genus and species name of the taxa that you're uploading. You'll be able to add extra information regarding kingdom, phylum, things like that through our taxon search with PBDB and NCBI. And if you're looking to add a little bit of images to your matrix of the taxa that you have and aren't able to look for the images, well, Morphobank will do the job for you with our connection to IDIC Bio and Encyclopedia of Life, where you just need to provide all your taxa names and it'll automatically populate pictures of your taxa if there are any associated with the name of your taxa. There's also additional functionalities like creating phylogenetic trees through Cypress, but all of that is just a large function of what Morphobank is. And then at the end, all the data is then published and made public in order for everybody around the world to access. Since it's open access, it's free to access. And so today I'll only be talking about the data upload aspect because that's the issue that we are currently battling and that's what the AI-powered solution is for. And so this is the data integration challenge that Morphobank is currently facing. And so the paleobiology database had about 650 phylogenetic matrices that they weren't able to keep up with on their website. And so they decided to provide these 650 matrices to Morphobank to make it a more robust data repository and sort of the only data repository that has all the phylogenetic matrices to make it an open access kind of concentrated resource for everybody to have. However, we've battled an issue that all these matrices that are provided by the paleobiology database have the taxa information, have the matrix information, but don't necessarily have the character names and character state information in them. So they're incomplete. So this kind of defeats the purpose of the findable, accessible, interoperable, and reusable aspects of FAIR data. And since we want to provide complete and accurate information as a means to reuse it, we're in the process of completing all these matrices before we ever publish them onto Morphobank. And so this is a layout of what a nexus file looks like. A nexus file is the computer name for a phylogenetic matrix. It's what encodes a phylogenetic matrix when you upload it to Morphobank. So I can go over the aspects of a nexus file now. So this is the taxa block right over here. It consists of all the taxa names that are associated with your matrix. Next up is the character names and character state blocks. So this will be where the character names and states go. As you can see, this block currently is empty, and this is how all the paleobiology database matrices look like for us right now. The character block is completely empty and it needs to be populated before being uploaded to Morphobank. And then at the end is the matrix block. Now, what we really want is the one that I'm pointing at right now, is where there's a taxa block. In addition to taxa block, there is a character block, but however, you can see that the character state labels have been completed in this matrix, and I promise you at the end of this Nexus file is a matrix block like this, which marks the file as complete. And so now that you have kind of an idea of what an incomplete Nexus file looks like versus what a complete Nexus file looks like, you'll be able to follow how this AI-powered solution actually works even better. And so before I'd go through the tedious procedure of looking for the empty matrix file, which was provided to us in a Google Drive folder, and then I'd have to look for the paper that's associated with this particular matrix. And that would take a little bit of while. And then the most tedious task of this was the data entry part, where I'd have to manually go in and type in each character name and character state associated with this matrix. And so matrices can easily range from 20 characters all the way to 1,000 characters. And let's say about a 50 character matrix takes 30 to 45 minutes to do manually. That's a lot of time, and it would have taken years to be able to complete the 650 matrix transfer from paleobiology database to Morphobank. So I came up with an AI-powered data integration solution. And so in the background right here in black, you can see the user interface for the AI that I've created. It's called the Morphobank PBDB PDF to Nexus file generator. You can see a preview of what a paper would look like that has the character names and character states. And at the very front is what I'd use to manually type in each and every character name and character state. It's a program known as Mesquite, which kind of helps you visualize the matrix on how it actually looks like versus the Nexus file, which is kind of just a bunch of lines of code. And so the AI-powered solution that I created takes in a document containing the character set and asks the user to provide information about the total number of characters and where these characters are located within the paper, so the page numbers. And then we use the advanced natural languaging processing capabilities of a large language model, an LLM, to analyze the document. We are utilizing Google's Gemini 1.5 Flash and 1.5 Pro at the moment, and the LLM intelligently understands the structure and context of the character list, even if it's presented in a complex format. As we all know, science papers can be a bit complex sometimes, and it's sometimes hard for me to understand them as well, but the AI is smartly able to sort of like unravel the mysteries of the scientific paper it's assigned to do. And then our app automatically populates the matrix file with the extracted character data. This means that I can say goodbye to typing it all out on Mesquite and focus on the quantity that I'm transferring over to Morphobank over having to tediously type each character, check if I've written the spelling right, because at the end of the day, we are looking for accuracy as well, because we want to provide true data to people who are able to reuse it. And so moving on, this is a little bit on how it works. We believe that, again, the solution is to use this AI in order to complete this transfer. And so harnessing the LLM power, the application utilizes advanced LLM language processing capabilities. Now, these capabilities are the fact that we don't have to train it for it to understand the scientific papers. It is so smart in itself that it kind of trained itself when we started using it on these scientific papers. And so after that, I've created sort of a complete toolkit where including text pre-processing for data cleaning, chunking for efficient handling of large data sets. We use count validation for accuracy and LLM evaluation also for quality control and accuracy checks. And the format of the output automatically updates to a format that's accessible to upload onto Morphobank, because Morphobank has a stringent kind of policy of what kind of Nexus file it accepts. And I'd also like to plug in the user-friendly experience that the AI is built around, so that the entire process of inserting the character state numbers and the page numbers are all kind of intuitive. And so I'm going to talk a little bit about the text pre-processing feature now, that trying to understand a complex research paper filled with tables, figures, and complicated formatting is pretty overwhelming, and so our application tackles this challenge by using intelligent text processing techniques. And as you all know, scientific papers have 2 columns and they're not read linearly, so we really needed our text pre-processing to not read things linearly. And so we made it in a way that it kind of reads the first column first, how we would instead of reading 1, 3, 2. The text pre-processing reads it 1, 2, 3, which is how a human would read it. And so this makes it easier to kind of extract the data in the right order versus it being in the wrong order and all jumbled up. We also don't stop there. We also apply sophisticated post-processing techniques to clean up the text, where we take out any unnecessary formatting or special characters and other extraneous things that would sort of confuse the LLM and throw off the accuracy of it. And so by delivering like a high quality clean text to our LLM, we are guaranteed like a nice high quality output. And then we're going to move on to what chunking and sliding windows are. So LLMs are pretty powerful tools, but they do have a finite attention span, just like me right over here. So they can only process a limited number of characters or tokens at once. This can be a challenge when analyzing long and complex research papers. And to address this, we use a technique called sliding windows. We break down the research paper into manageable chunks, each within the LLM's processing capacity, so it's not overwhelmed while it's reading these characters, and each chunk is given to the LLM to extract a certain section of the characters. And so talking about the validation methods that we use for accuracy, the response of an LLM for each chunk is validated, and we ensure that it's done in a 2-step process. One of the steps is pretty easy, while the other one is a more complex step. We can check the number of characters extracted by our LLM against the number of characters that were kind of input in that little state the number of characters that are in your paper entry. And so this helps us catch like any duplicates that might have happened or any missing sort of characters in number. However, we don't stop there. We use a separate LLM to model assess the overall quality and correctness of the extracted data. And so this LLM acts as a second pair of eyes, ensuring that the matrix is sound and accurate. And so any failed chunk in these kind of count validation as well as the LLM evaluation is then reprocessed. And so this 2-pronged approach gives us the confidence that the results generated by our application are accurate and reliable most of all. Lastly, you must be thinking that this must be pretty slow if it does it linearly, and talking about all the chunking that's happening and there's limited processing capabilities in an LLM. But LLMs are slower than traditional computer programs. Now, this is contrary to popular opinion, or like opinion that's normal, that, oh, LLMs are fast, they're great, but they're actually pretty slow. And so we make use of a parallel API calling for LLM to reduce the time required for data preparation. This way, each chunk of text is processed parallelly instead of linearly. And so think of it as having a team of workers all working on different parts of a project at the same time. This parallel approach allows us to achieve like 10 times, even 100 times the speed compared to a manual human like me doing it. And so this could possibly be the future of evolutionary research, because becoming a part of all these workshops that Morphobank has hosted, I've realized that people do have empty matrices that they need to fill, empty nexus files that only have the taxa block and the matrix block. And I've been able to put them into my AI-powered tool and have output complete nexus files which were then uploaded onto Morphobank for the public to see, which is a great deal because it's a great campaign towards having more fair data in the scientific realm.
Okay, I'm going to cut it off there just to keep time. Our next speaker is Caleb Charpentier, and we'll get started.
Hi, everybody. My name is Caleb Charpentier. I'm starting my 3rd year of my PhD program in Josef Wetter's lab. He's over there at Virginia Tech. And the broad sort of theme of my research is that I'm interested in how we can use deep learning and AI methods to add some biological realism, or hopefully increase our biological realism for the way we measure morphological traits, primarily from images. And before we get to all the complicated AI stuff, I think it's a good idea to revisit the fundamental idea of what we're doing when we measure things to begin with. And this is a process that everyone as a biologist is familiar with. For example, if I'm interested in measuring these fish, there's some relationship about these fish that I think is interesting. So I might be measuring the size of the fish. As you see, as you go from left to right, these fish are increasingly getting bigger. And if I think that sort of ordinal relationship between the fish is important or interesting for whatever process I'm trying to model, I might create some sort of transformation process from those real fish into numbers, whether that's by just laying them next to each other or using a ruler, such that the relations between the numbers hold that relation that I think is interesting. So in this case, each of these fish is assigned a number, where the order of the numbers maintains the order of the fish. And you can do this for different sorts of things you might want to measure. In addition to just ordinality, you might think of ratios. So I think 2 of those green fish are proportional to the big fish on the top. And the 3 small ones on the bottom are proportional to 1 of those. And so I cast them into numbers such that relation holds true for groups of fish or for individual fish. So the green fish is half the worth of a red fish. Now, over the past couple of decades, there's been this sort of phenomics movement where a lot of the idea is to just measure as many things as we possibly can. And this has been done in a lot of ways. For example, we might want to measure landmarks. So we have some scientist somewhere decides the thing that's interesting is these homologous landmarks, and they create some pipeline that can mass produce landmarks for all sorts of specimens. It can be landmarks, it can be things like segmentation if you want to measure areas of volumes of surfaces, or things like color patterns. And as you might see, in most of these phenomics cases, they're using sort of a traditional idea of measurement, where some human researcher decides what is cool, what is interesting, and they prescribe some method for converting that into numbers. But sometimes this can be a bit limiting, this human way of doing it, because humans tend to measure things in a way that's rather both redundant and incomprehensive sometimes. So as an example, one trait that's measured in all sorts of systems, from fish to plants, is aspect ratios. Aspect ratio is just the height ratio to the width of something. But as you can see, this this oval and this rectangle have the same aspect ratio, and they're very different shapes. And a square fish is going to have very different hydrodynamics than an oval-shaped fish. And so if I want to measure the outline shape of a fish, I might take aspect ratio, but then add a whole bunch of other variables that can describe that. But the issue now is I have measured 20 things that are just the aspect ratio of the fish. And so you might find patterns that seem important, not necessarily because there's anything fundamentally biologically interesting going on, although there might be, but because you just measured the same thing 10 times. An attempt to get around that in recent years has been to use these sort of latent phenotyping methods, where the idea is to learn some underlying variation describing these overall patterns. One example of this is PCA, but a neural network technique using sort of modern neural networks is called an autoencoder. This diagram you see on the screen is an autoencoder, and what it's trying to do is learn an identity function. So as you can see, this Arabidopsis picture on the left goes into the network, and the network tries to give you the same picture back. But it does it in such a way that there's a bottleneck in the middle. So as you see in the middle, that whole image is represented with just 2 numbers. And so the idea is that if you can find a few numbers that summarize this entire image, maybe those dimensions in some way can capture the variation that you think is important. But the difficulty with this is I don't know what those numbers correspond to. I don't know how to do biology with that, and there's a good chance that it's overfitting. And for the sake of this talk, we're going to call that, sorry if my volume's going in and out. For the sake of this talk, that vector is called a latent or embedding vector. Those are the terms that are normally used. Sometimes they're used different meanings. We're going to use both of them as the same thing. I like to summarize this problem of neural nets overfitting and being black boxes and humans measuring things in sort of inconsistent ways as a contrast between 2 quotes. The first quote is from Huel et al. In 2010. And it goes, "Rapid progress on any biological problem rests on the hope that there is at least one viewpoint to each problem that makes causation relatively simple. I would generally agree with that if you have a really, really overly complex model. I don't know how to interpret that. On the other hand, we have this quote from H.L. Mencken that goes, for every complex problem, there is a solution which is clear, simple, and wrong. And I think at first hand we would agree with both of those things. We need simple models to do science, but also there are a lot of wrong answers, right? So how do we get around that? And so the sort of contrast that Whewell states that Mencken doesn't is causation. I might need to go back one more time. Is causation. We want problems that are simple but are also consistent with a biology that we understand. Sometimes you might choose a more complex model over a simple one if you know that model represents something that you already know about the world. And so going back to the initial problem, these high-dimensional phenomics measures can still sometimes be prone to redundancy, ascertainment bias of us repeatedly measuring things we think are cool, and incompleteness, because we measure a bunch of things, but a large portion of those things are sort of the same thing. These latent phenotyping methods try to address those problems, but they're really difficult to interpret, and they're difficult to evaluate. If I don't know what this is doing, how do I tell if it's doing it right? So a solution for this is what we call knowledge-guided machine learning. And knowledge-guided machine learning is more of a philosophy than a particular model. The idea is when you evaluate the performance of the model, you should consider the model's accuracy, its simplicity, and its causal consistency. And we're very used to doing the first thing, right? Everyone needs their model to be accurate or it's not useful. And we induce simplicity in various ways. We might use sparsity, we might use information criteria like AIC or BIC to prefer simpler models. In such a way to try to make fewer assumptions. But we don't necessarily always explicitly think about the causal consistency. Like I said a second ago, sometimes you might prefer a more complex model over a simple one if you know that complex model is representing something about the world that you think is correct. But how do you actually do this with a neural net? It's just a big pile of numbers. One possible way, and it's not the only way, but one possible way is to incorporate knowledge of whatever your experimental structure is into the model's architecture. So there's this study from Ubens et al. In 2020, and they breed plants. I'm not a plant person, but I like this example. And so basically what they did a very simple experiment. You have two populations of Arabidopsis. One population you subject to some sort of treatment. It can really be anything. It's a very general method. One population you don't subject to that treatment. And after the treatment, you take images of individuals of the population in a time series. You might have 5 images of the same plant throughout its life. And you use what's called a long short-term memory network, which is an autoregressive neural network for compressing time series, to basically say, this is a representation of that plant all in the bottom throughout its entire life. This is a representation on the plant on the top throughout its entire life. And then you say, try to find a vector that can distinguish those two groups. You don't tell it what the phenotype is at all. It just tries to figure it out from the image. So using the autocoder like we said for earlier, you get those embeddings in the middle that are now sort of reflective of the experiment that you did, and you can use those numbers for downstream processes such as GWAS. In their simulated case, where they simulated images of Arabidopsis, they were able to identify the QTLs they simulated that distinguish the populations without even telling it what the traits were. It just fed it the image. Not everyone does that sort of biology, though, where you're doing just hands-on experiments and tweaking things. Sometimes you might be interested in something like a phylogeny, which is still sort of a causal structure of change. I'm saying there are differences between these groups because this thing happened, whether I induced it or because evolution just split these groups up. So how can we incorporate other structures, such as phylogenies? Well, this study by Elhamad did it. Elhamad et al., including myself. Where basically the idea was what we wanted to find embeddings of images. These are a group of cyprinid fishes from the Great Lakes. Where what we wanted to do was we wanted to find information from the image that was reflective of phylogeny. So what we did was we split the image variation instead of into one vector, we split it into 2 vectors. Ignore this part with a transformer right here. It's not important for what we're talking about. We're interested in ZP and ZNP. ZP is the embedding that is reflective of phylogeny. And in sort of simple terms, the idea is we're trying to find an embedding that maximizes phylogenetic signal in some way. ZNP is trying to minimize phylogenetic signal in some way. You can concatenate those 2 vectors and rebuild the original image with all of the information, but now you've sort of pieced apart which things are interesting to an evolutionary biologist and which aren't. And when you see, if you look at the little heat map in the top corner, when you get distances between those embedding vectors, these are the distances that the model found, and these are the actual tree distances from the phylogeny, and while they're fairly different, the broad patterns are still pretty good. So now that you've seen how we can sort of incorporate our knowledge into these models, how do we assess whether the models are working? You can sort of do it in the way we already assess models. And I break this down into two pieces. You can assess the plausibility of the model, and you can assess the performance of the model. So can this model work in any world? And is this model actually working for this particular thing that I'm trying to figure out? To assess the plausibility of the models, we can also do something we can already do, which is simulate data. And in the case of these autoencoders, we need to simulate image data, which doesn't sound like a trivial task, but there are actually a fair number of ways to do this. One way to do this is with a pipeline I created called Trait Blender. It's built on top of Blender, which is 3D modeling software. And using Trait Blender, if you have some sort of theoretical morphospace, you can simulate trait values in R using phi tools or something, convert that into a 3D model in Blender, and generate data sets of tens of thousands of individuals or whatever you're interested in, adding random noise, variation, whatever you want, where you know the entire evolutionary process that generated those 3D models. In this case, you know the tree. Because you made up the data, you can know other things as well, such as the ancestral states, if that's the thing that you're interested in. These are some little fun monkeys that I like to play with as a really simple case. But as you can see here, we have this map where we know the entire history of these 3D monkeys. We can even make pictures of the ancestors if we want to. If we want to assess how well the model is actually performing when I want to do some experiment with some real data where I don't necessarily know the ground truth, you can do this in a variety of ways. If you're looking at an evolutionary study, you might want some metric of phylogenetic signal on the embeddings. You can do tests of population divergence if you're doing some sort of study on that scale. There are various ways of sort of permuting your tree where you can sort of remove branches on the tree, train the embedding network, and then see if it places the missing clades in the right places. You can look at evolutionary distances like we did in the shown study. And you can look for correlation with known traits. You can see, are these neural network models sort of agreeing with what biologists already think? So the broad sort of takeaways that I would like you to take away. These latent embedding methods can address some issues that traditional character construction sometimes struggles with. But they're hard to interpret. Knowledge-guided machine learning can make them more interpretable if what you're trying to interpret is the sort of process by which this variation changes, which is a lot of what science is. And once you have these interpretable models, they can be tested in similar ways to the ways we already test models via simulations or using various summary test or statistics. And with that, thank you to all the people. That's my lab. That's our virtual postdoc, Brian, looking over the mountain.
Hi, everyone. Thanks for being here. Thanks to the organizers for putting together this fun symposium. My name is Patrick McKenzie, and I'm a postdoc in Robin Hopkins' lab where I usually study flowering plant population genomics and speciation genomics. But today I'm really excited to talk to you about something very different from that, which started as an iNaturalist passion project and has since turned into something that's made me really excited about a fundamental signal in North American flowering plant phenology. And so, of course, we have to start by just marveling at the diversity of floral morphologies that we see in nature. And so lots of previous research has gone into understanding the evolutionary processes that drive the diversity we see in flower shape, size, color, aroma, and whatnot. And in doing this research, plant scientists have found that a lot of this morphological diversity can be decoded to associate certain floral morphologies with specific pollinators. And so this is known as a pollination syndrome, which is just describing the convergence of a suite of floral traits on the preferences of pollinators. And so throughout this talk, I'm going to focus on one specific pollination syndrome, that of the hummingbird flower. Hummingbird flowers are typically red. They're typically tubular. These are things that are designed that tend to attract and specialize on hummingbirds. And they also have a suite of traits that deter less efficient insect pollinators. And usually with hummingbird flowers, you kind of know it when you see it. So the top row here is showing you flowers that are very bright red and orange and very tubular. And then on the bottom row, we see flowers that are closely related but insect pollinated relatives. And so while this hummingbird pollination syndrome has been well characterized for a long time now, our access to large-scale occurrence data has been a lot more limited when a lot of this data has been collected than it is today. And so we set about studying whether we could leverage new large-scale occurrence data to possibly reveal new patterns in the ways hummingbird flowers might try to specialize on hummingbirds. And so the specific large-scale data I'm using throughout this talk comes from the participatory science platform iNaturalist, where any one of us can walk around and take pictures of the things that are living around us, and then scientists can use those observation data to learn about biology. And so when we looked at this data, one particular signal stuck out to us. So here we have two maps moving through the first 20 or so weeks of the year. On the left side, we have the occurrences of all plants in our data tagged as being in flower. And on the right, we see hummingbird occurrence. And so the signal that really struck us is that for a window of time in the spring, flowers are blooming far up the East Coast and hummingbirds have not arrived yet. And so this is like a key slide for interpreting the rest of the talk, because it leads us to ask the question that if red flowers do associate so closely with the arrival of hummingbirds, but hummingbirds aren't there yet, or if red flowers associate so closely with hummingbirds, but hummingbirds haven't arrived yet, should we expect red flowers to be depleted in this area at this time? And so to kind of rephrase this question more generally, in the context of pollination syndromes, we're wondering if we could, in some cases, consider flowering time to be a pollination syndrome trait. And so in our case, if so, we expect that the flowering time of red flowering species would correspond to the observations of hummingbirds. So to get into this, we started by downloading all iNaturalist observations for our broadly North America, it's the United States plus or minus some on South and North, labeled as flowering angiosperms. And this gave us a data set of about 1.7 million occurrences of over 13,000 species. And then all we had to do was map each observation to a flower color and then analyze the occurrence of red and orange flowers versus the arrival of hummingbirds. Easy. Actually, the second step is really hard. We identified 3 possible paths that we might take to associate flower color to flower occurrences, one of which would be to just go through this data and manually label each flower with a color. Obviously, that's not possible for us. We also explored using a trait database, and we were pretty happy finding that we had flower color data in this database for over 2,500 plant species that we could map onto our occurrences. But our denominator here of 13,000 species is still pretty large, indicating that there's a lot of data that we're not covering here. So that's when we turn to computer vision, which is my relevance to this symposium. Our idea here is that, so in iNaturalist, each unit in the iNaturalist taxonomy is assigned a default species photo. And for angiosperms, or flowering plants, that default photo is almost always that plant. It's like a beautiful picture of that plant in flower. And so we thought we could extract information from the default photo for each species. And so to do this, we use the OpenAI developer platform. They have an API that interacts nicely with Python. And so we first just queried the link to each of the iNaturalist default photos for each taxon. And we passed that link to the GPT-4 with vision model to label the flower particular color category. And so yes, this is the same GPT-4 with vision model that you are probably interacting with day to day through ChatGPT. And so we had a lot of success with building a complete data set this way. So we start with over 13,000 species, and after doing this color labeling protocol, we end up with just under 12,000 species that we were able to label correctly. And that maps onto almost 1.7 million occurrences of plants that are in flower and are now color labeled and tied to a GPS point and a date. And so we're really happy with our kind of species coverage here. And I also wanted to touch briefly on the fact that we did validate this data. Each of us co-authors, we manually labeled a 250 species subset, And among ourselves, we found we agreed on about 90% of our color labels. And we found pretty similar rates of agreement with the labels assigned to those species from our trait database and from GPT-4. And in particular, actually, GPT-4 did pretty close to our human error rates and appears to give us more accurate results than the trait database does. And so to get into what this data actually looks like, I should start with some basics. So we're here looking at the breakdown of the frequencies of the different color categories assigned by GPT-4 with vision in the United States. In the left plot, we have the breakdown by species, and in the right plot, we have the breakdown by occurrences. And so there are two major takeaways here. One is that there's really high concordance between these plots. So occurrences are recapitulating species diversity here. But also, we have 4 colors that are taking up over 3/4 of our flowering plant observations: white, yellow, pink, and purple. So yes, this is a very simple result, but this is a very simple result that's very hard to generate without this kind of really large scale data. And so now the rest of the talk will focus on the initial question about red flowers versus the other colors. And to start, I'm going to revisit week 11, where you might remember we looked at the slice in time when flowers were blooming and hummingbirds weren't there yet, and asked if red flowers would be depleted. Well, we can see this now. Very clearly, yes, red and orange flowers are not present in our data at this time, but all we have to do is shift ahead 10 weeks to week 21, where hummingbirds have now migrated up the eastern US, and look at those same frequencies, and we can see that now no red flowers are no longer depleted. We actually see them present in roughly equal proportion to their overall frequency in the data set. Our second vignette of 3 total is to look at the leading edge of the range of each color in our data set, while limiting our perspective to the eastern United States here. And so to quickly illustrate what we're trying to do here, just consider the range of one flower color. And as the day of the year advances along the x-axis, we can track the northward extent of the range of that flower color. And so we're keeping track of the northern latitude of this range. And so if our data here matches our hypothesis, we should see that the northern extent of the red flowers, kind of across the x-axis, lags behind that of other colors for any given day of spring. So here are our non-red and orange colors. And we can see that, yes, there's a very clear signal of this pattern of red and orange flowers lagging behind the other flower colors. Our third and final analysis was to explicitly study the inferred spatial distribution of our different color categories. And here we're using species distribution modeling with Maxent. And for simplicity, we're just looking at red flowers versus white flowers, and we're looking at sliding windows of SDMs through the year. So this is kind of the main result. I'll let it go once or twice so we can see the colors creep northward. We're ticking through days of the year, and you can see that red and white flowers are both advancing northward through North America, but especially early in the year, so days 30 through 70 or so, white flowers clearly lead red flowers in their flowering time. We can condense this into just one plot where we're basically subtracting the left side of that previous slide from the right side, and we're using blue to map areas where white flowers are leading red flowers in their flowering time. But we can get even more out of these species distribution model results by looking at variable importance. And so this is now where we can go from observing our pattern of red and orange flowering later than the other colors and actually associating it with the pollinator side of things. And so here we're rerunning the same distribution models for white and red flowers, but we're also throwing in hummingbird and generalist bumblebee pollinators as additional predictor variables and looking at variable importance through the spring on the x-axis. And we can see that through the spring for white flowers, bumblebees come out as being very important predictors of white flower distribution. And in contrast for red flowers, hummingbirds are assigned very high importance as predictors. And actually, to find that same signal, we don't even need fancy distribution modeling. We can look at the raw data. So here, I'm using the same kind of mentality we used before, looking at the leading edge, but I've also added on the trailing edge we have red flowers on the left and we have white flowers on the right, and then I've overlaid the hummingbird pollinator on the left and the bumblebee pollinator on the right, and you can see broad overlap in both of these plots. And in contrast, if we flip these around and pair hummingbirds with white flowers and bumblebees with red flowers, we don't see nearly as much concordance. So, something I have consistently loved about this project is how simple it is. We have 3 main big conclusions. One is a resource that we're going to share with the community, which is this big mapping of species to floral color traits. But we also have this newly described pattern of red and orange flowers lagging behind other colors, as well as a pollinator association specifically with red flowers and their specialist hummingbird pollinators. And so taken together, we think that these simple conclusions demonstrate some correlatory evidence of hummingbird-driven temporal constraints on flowering time, which is adding to the already complex ways that we know that flowers might interact with their pollinators. So we have a bunch of downstream things to look at with this type of data. We want to look for related signals in other seasonal pollinators. I think it'd be really cool to break down our data into species abundance and look at relatedness, as well as within species variation, which we haven't done in this analysis. And then, of course, trying to understand biases in our GPT-driven data extraction method. But in the meantime, if you're interested in more kind of on this subject, I saw that Chris Talbot is speaking at 3 p.m. today and is using some very similar data, but is taking kind of a phylogenetic lens on the problem. So I suggest going to his talk because it looks like it's going to be really cool. And with that, I would like to acknowledge my co-authors, Dr. Robin Hopkins and Dr. Andrea Berardi, as well as the whole Hopkins lab and everyone who's given me helpful feedback. And then, of course, the tools that made this possible, which is iNaturalist and its community of users, and OpenAI and its GPT model. Thank you.
Okay, thank you so much, Sarah. Yeah, I'm Claudia Solis-Lemus. I'm an assistant professor at the University of Wisconsin-Madison. When I originally submitted this talk, I wanted to talk about phylogenetic networks, as is the original title that you see in the session. But then I somehow ended up in the generative AI session, and I also have things to say about deep learning. So now I'm going to try to talk about both in 15 minutes. Okay. So I'm going to start with networks. So Cecilia and I developed a method to infer phylogenetic networks that are an extension from trees that account for hybridization. And this is a pseudo-likelihood model that was implemented in a Julia package, phylo-networks, and the method is called SNAC. It's the most scalable version to date to infer phylogenetic networks because most of the other approaches rely on you evaluating likelihood on full networks. And this is a divide and conquer approach. You only evaluate likelihoods on quartets. So it is much faster. And I want to give a brief overview of the type of network methods that we're still working on in my lab. First of all, Kevin Kong is a postdoc working with me. He gave a talk on Saturday in the network session where he was talking about how we're extending this problem from level 1 networks. We are networks where we can, the hybridization cycles defined here in the network, they cannot intersect. That is a level 1 network. Kevin is extending SNAC to the case of level 2 networks, like the one is shown over there. So it's the case where hybridizations or recipients of gene flow can further hybridize. So it's a more complex network that we can now try to infer. And then Nathan Colvo is a PhD student working with me. He also gave a presentation on Saturday about there is a bottleneck of how big of a network we can infer. Because of this, we have to search the space of networks to maximize some criteria. So we cannot really go to really, really big networks. The only way we can get a network with like 1,000 taxa is if we break up the taxa into subsets and then we merge the subnetworks together. This is what Nathan is working on in a method that's called Infinite. And I want to highlight 2 other network stuff, and then I will switch to AI, which is I know what we all want to hear about here. So Bella developed a tool to infer or detect these hybridization cycles in the data without having to optimize in network space. So other criteria like likelihood approaches, like I said, you search the space of networks until you find the optimum. Bella's work relies on polynomial equations called phylogenetic invariants. So you just plug in your data into the invariants. If they vanish to 0, they point at which is the hybridization cycle that best fits with your data. So there is no optimization step. It's simply evaluation of polynomials, so it's ultra fast. We even dare to call the paper ultra fast learning because it is actually very fast. It's just evaluating polynomials. And there is a Julia package for that we're just developing, which is PhyloDiamond. And then the last one, the last project on phylogenetic networks is we're finding ways to embed networks into some mathematical space This is work in collaboration with Julia Palacios in Stanford, where we can represent a ranked phylogenetic networks as a matrix. Julia has done this work for ranked trees already. We are extending to the case of networks. Once you have a network in a matrix form, which is not the adjacency matrix for those that are familiar with matrices, you can do standard statistics like clustering, calculating mean, calculating variances. Anyway, this is the overview of phylogenetic networks that is happening in my lab. But now I can switch to what we really want to hear more about in this session, which is machine learning. I want to start by saying that I'm a statistician. So I was trained as a more classical statistician, so I didn't want to get into machine learning. I was like, no, that is not for me. I really just want to do likelihood-based methods. That's, that was my area. But there is this nice pressure from students that want to do deep learning, collaborators that want to do deep learning. Everybody wants to do deep learning, so you're like, okay, well, let's do deep learning. So the very first projects that we started working on deep learning were very classical classification problems or prediction problems. So in the first example on the soundscapes, we're collaborating with the Burivalova's lab, where we have sound recordings from rainforests. And from the sounds that are treated as images, like spectrograms, we want to predict the biodiversity index of the rainforest. This has huge implications for conservation, because then now you can have an automatic way to know the health of the rainforest. This is very simple neural network that is taking the spectrograms and just pretty much predicting number of species and how diverse the rainforest is. The second project is on predicting antibiotic resistance from bacterial strains. The input in this project is genes, genomic sequences, different type of input data, but again, we're trying to predict a binary class, resistant to antibiotic or not. Simple neural network. And the third project is predicting yield or disease susceptibility for potato crops. And in this case, the input data is soil, microbes. So we have relative abundances from microbes in the soil and other characteristics from the soil. Again, it's just a neural network. Actually, that's a lie. For the potato yield disease project, we use random forest and Bayesian neural networks. But these are, like I just said, just classical classification tools that they are very, very known in deep learning. But what about phylogenetics, right? How do we do deep learning within phylogenetics? And there was this really interesting paper in 2019 that they were trying to predict or estimate a quartet, which is a tree with 4 taxa, from sequences. So in this paper, they train a neural network model where you input 4 sequences, and it chooses which is the quartet that we're coming from. In this case, when there are only 4 species, there are only 3 possible quartets. It is, again, a classification task. So we're choosing a label between 3 labels. Okay, and we're training this model. They show in the paper, in the table here, that their model outperforms standard phylogenetic methods like likelihood, neighbor joining, Bayesian, especially in the case of long branch attraction, which is the one that is shown here. When you have 2 sister lineages, one has a very long branch, the other has a short branch, some of the traditional phylogenetic inference methods will mismatch those lineages. But there is a caveat with this model. There's a limitation with this. To my knowledge, this was one of the first methods to use neural networks to infer trees. But it has a limitation that it doesn't account for tree symmetries. If you look at the quartet here, if I swap 3 and 4, it's the same quartet, right? Or if we flip it, and we have 3 and 4 on one side, 1, 2 on the other, it's the same quartet. The model doesn't account for those symmetries. The only way it's considering those is expanding the training data set by including all permutations of the sequences. So you have your 4 sequences, 1, 2, 3, 4, and then you have 24 possible permutations of those 4, and you use those 24 for each simulated data set as training. So they are accounting for the symmetries, but by artificially increasing the training samples. And I was talking to Leo. Leo was an assistant professor in math in Madison, and he is the expert in deep learning. Like I said, I'm not. He developed this method, this new neural network model that is intrinsically accounting for the tree symmetries. So it is intrinsically invariant to permutations to the sequences, so you do not need to expand your training data set because the model is accounting for those permutations internally. This is not an improvement in accuracy. In fact, if you train them both, they were equally accurate. But we think it's an improvement on the sophistication of the model and then also in memory. We prove that as well in the paper. And then Shudong Tang extended this model for the case of 5 taxa. And the sad news is that it never outperforms maximum likelihood or neighbor joining. So you spend hours training the neural network, and then you don't outperform traditional phylogenetic networks. But still, it was an interesting model. I think it made sense at the time. And this is a slide that I stole from Zhang's presentation. He was in Evolution last year, where we think that perhaps classifying or a classification model is not the right way to go in phylogenetics, because if you have 4 taxa, like I said, there are 3 labels, okay? If you have 5 taxa, there are 15 trees, so those are the labels you're choosing from. If you want to do 10 taxa, you have over 2 million labels. So you cannot train a model to classify among 2 million labels. So maybe classification models are at that end. That's a very strong statement for phylogenetics. But I'll tell you what we have been working on. In the meantime, as we were exploring models ideas, there have been many other applications of deep learning in phylogenetics. I want to highlight 2 of them that are like really, really good models. So there's Megan's work on adversarial networks, neural networks, where there are trees that simulate data, and then you see whether the simulated data is indistinguishable from the real data. And that will point at the tree that best agrees with the real data. And then there's Phyloformer that what is predicting is pairwise distances. So you predict the pairwise distances, and then you use a distance-based method to get the tree. These 2 are very sophisticated, very good deep learning models, but they're not input-output models. By input-output, I simply mean we would like something that we input the sequences and we get a tree without any intermediate step, everything we did in deep learning framework. And that's what Zhang really wanted to focus on, and he thought the answer was in generative models. A spoiler, it's not, or at least we couldn't find it. But the generative models is when you have a set of training samples and you're trying to learn a distribution, so that with new data, you just get from the distribution the tree. So it's a different type. It's very widely used in other graphical structures, like social networks. They all generate a network. So we thought it would make sense for phylogenetics to try it in phylogenetics. Zhang worked really hard. He was trying one-shot models that are based on graph attention networks, where this is an autoencoder, like the one Caleb was talking about. I don't know where he is. So where you're trying to find a latent space for each node in the tree, and then try to predict whether there should be an edge between the trees. So you start with a tree, and then you reconstruct the tree. The challenge for phylogenetics that I don't know if others have seen this as well is that if you try to bring ideas from other graphs structured like social networks, for example, they have data in every single node. Even if there's missingness, there's expectation that there's data. In phylogenetics, we have a very clear missingness pattern because we only have data at the leaves. We don't have data in any of the internal nodes. And we have ways of imputing that data. We can estimate what is the ancestral sequence. It turns out that, at least for some of the methods we tried, that doesn't work because you are not gaining new information about the neighboring structure. You are using the leaves to put the data in the parent node, so you are really not learning anything from that. So the one-shot model, we couldn't make it work because we believe, this is our hypothesis, we believe it was the lack of data in the internal nodes. And then he also tried what sequential models that have been used in molecule generation models. Basically, you break up reconstruction of the tree as a set of binary tasks. Should I join these 2 nodes, yes or no? Each of these layers is a separate neural network. So it is very hard to tune, because they are like this compounding of errors, and it was a lot of work to tune them. So one of the things that, we couldn't make this work, as I started saying. We couldn't make it work. And again, this is the bitter statistician. We were like, okay, the traditional tools work to build trees. Like, we really need to go down this path. I'm not trying to tell people not to do that, but I don't think we will necessarily pursue this path unless, but first of all, Zhang graduated. So I will need another student that is motivated and interested to keep trying. So if anyone's interested, please let me know. But for now, we couldn't find this input-output model that we were looking for. So sorry for the bad news. But there is one area where I think machine learning is very good for phylogenetics, and that is the case of embedding. So Caleb was already talking about this. And there is this paper, Ding's paper, where they have a variational autoencoder. And here, there are no images. So in Caleb's talk, there were images. Here, the input are sequences. But you still embed them into this space of just 2 dimensions. Your latent vector, which for me feels like black magic. Like, you have this sequence that is just so long, and you're representing it with just like 2 numbers that is capturing all the information somehow. But it seems to be quite interesting if you look at the plots on the far right for you. This is simulated data. You see the nodes colored yellow, green, on top of purple. The leaves, it has a very interesting geometric pattern, the latent space, because it has like this cross shape where it starts at the leaves, and then you start working towards the center to get the ancestor. So there is something in the latent space that is being captured, some phylogenetic structure that is being captured. So we found this very interesting. That paper was simulated data. This is real data. It has the exact same structure, and you can actually embed the tree in the latent space, which we think is really cool. So this is the last slide. We're working on this. We're using this latent space to try to reconstruct ancestral sequences. Anyway, I wanted to promote seminar series and Julia File organization, but I don't have any time. Thank you.
So good morning, everyone. Thanks very much for the invitation, and it's my pleasure to present some of our recent works on autogression models for phylogenetic inference. So this is it. Okay. This is a joint work with my student Tian. Okay. Great. So maybe you guys are very familiar with phylogenetic inference. So I'll go through the first slides very quickly. So it's about a reconstruction of the evolutionary history, given some molecular sequence data like DNA, RNA, or protein sequences. And this idea of using tree-shaped graph to describe the evolutionary history dates back to Charles Darwin, and it has many applications in modern biological applications, and also some medical applications like B-cell evolution and pandemic things. Okay. So in practice, people actually cares about uncertain quantification, so that's why Bayesian approach are usually preferred. And the setting is kind of standard. You have a likelihood function, which is due to false distance. You can evaluate the likelihood very quickly, and then you assign some priors for it, and the goal is to estimate the posterior. Due to the discrete nature of the phylogenetic tree topologies, currently people are just using this random walk MSMC, which turned out to be not that efficient, and I will describe some reasons for it. First one is that...
Can you speak out of the microphone? Yeah, go ahead.
Okay, thank you.
Thank you. Wait, let me make sure it's on. Yeah.
So why I didn't see the slides here? Okay, good.
So try that one. Is it working?
Yeah, it's working. Okay, it's working. Okay. So the first thing is we have a very large searching space, right? If you account for the total numbers of unrelated trees with n taxa, then the number is 2n minus 5 double factorial. That's combinatorial exploding. Another thing makes it even worse is that the tree topology and branch length and many other parameters are kind of intertwined with each other. So you update the tree topology, maybe you don't know how to update the branch length, for example, and that makes MSMC has very low acceptance rate, right? And then that makes it hard to scale to data sets with many sequences. Okay. So in the past, few decades, people have realized that actually the bottleneck really resides on exploration in the tree topology space. So we have developed many ways, like guided tree proposals for Bayesian phylogenetic inference, pioneered by Largate and Hona and Drummond in 2012. And then later on, this is called conditional clay distribution. And later on, we extended into the so-called substituting networks for probability estimation task also, and also for the variation of Bayesian phylogenetic inference. You can take a look at those papers if you're interested. And then there's recently a machine learning-based algorithm or ideas for designing guided tree search algorithms for maximum likelihood estimation. That is pioneered by Azuri. Okay. So all these approaches actually propose conditional or unconditional distributions over tree topologies via some kind of heuristic features. Okay, that depends on that requires substantial domain knowledge and may limit those their expressiveness, like it's only in a confined domain. So the question is, can we design some powerful probability models over the tree topology space? Okay, that is going to help you to design more efficient explorations in the tree topology space. So that's the motivation or starting point of our work, which we call the AR tree, that describes an autoregressive model for tree topologies. And in the deep learning community, actually, this is a very common idea. And they're commonly used to model sequential data. So we can decompose the joint distribution in a very high dimensional space by factorizing it into a series of conditional distributions. And you can take a look at all those examples here. So in our work, we actually devise a similar thing, but it's for phylogenetic topologies. So it's based on our recent work on learnable topological features. In a nutshell, you can see this kind of formulation here. So we're not directly describing this autoregressive model over tree topology space. Instead, we're describing this autoregressive model on a decision sequences. And we find a connection between the tree topology and the decision sequences, which I will describe in the next slides. Okay? So that's why we can propose this autoregressive model. The idea is actually pretty simple. So each tree can be viewed as a sequential generating process. Starting from the first 3 taxa, you have a unique tree, right? And you're gradually adding one taxa by another taxa by attaching to the branches. So every time you have a current tree topology, you start to select a random, select an edge, and then you attach the new taxa to the edge, and you can repeat this process until you recover this whole tree topology. Okay? So during this process, actually, you can transform a tree topology into a decision sequence. That is the edges you selected during this selection process. Okay? And then once you have done with this kind of decision sequence representation of the tree topology, you can easily prove that there's a one-to-one mapping between the tree topology and this decision sequence. Okay? And due to this kind of representation, now we can describe a probabilistic model over the space of tree topologies by doing that in the sequence space. And the sequence has a sequential decision this structure. So that allows you to construct a autoregressive model of it. The only thing is how to model this condition distribution, right? Okay. So to do that, we use a learnable topological feature, which is also our previous work, to parameterize these conditional distributions. So as the previous presenter actually mentioned that for phylogenetic model, there's a very difficult thing to deal with. That is, we only have the information for the tip nodes, right? So if you want to use this graph neural networks things to parameterize the conditional distributions, first thing is to find an embedding for the interior nodes. Okay? And we managed to do that using Dirichlet energy minimization. Due to the time limit, I'm not going to describe this in details, but if you use Dirichlet energy minimization, you'll be able to find some very intuitive embedding for the interior nodes. And based on that, you can do message passing. Okay, that's the core component for graph neural networks. And it contains 2 steps. One step is to aggregate messages from the neighboring nodes. So this is called ask message. And once you get these messages, you aggregate the messages from the neighbors to form your new node feature. And this is called updating this layer. And this one is required to be permutation invariant. So because it acts on a set. Okay. So once you do that, then this above procedure is called the graph convolution operator in GNNs. And in our implementation, we just use a very simple special one called edge convolution operator, proposed by Wang et al. In 2018. Okay. Another thing is we have to describe a conditional diffusion given all these previous decisions, right? So in order to do that, to capture this previous decision information from previous decisions, we use a recurrent neural network architecture. And this is kind of standard. Every time you just keep the current hidden states of the nodes. And then if you using GNN to aggregate information from the neighbors and get a final node feature for the current time step FNV, you try to aggregate this information into this latent representation by using a gated recurrent units. This is one of the popular choice for RNN constructions. It's called GRU. And then that will help you to update the latent representation into HNV, the current time steps. and you can do a zero initialization for this latent states. Okay, and we still have a very special case to deal with. But that is every time you update the tree topology, you add a new text to it. And we're using this Dirichlet energy minimization to get the initial embedding. And that will change again, because every time you redo this energy minimization, then you will get a new set of initial features. And that is different from this traditional graph orthographic models, because in that case, the nodes has a fixed feature, but now we have a dynamic feature. So to deal with it, we actually introduce a time-guided readout step. That is, we combine this latent representation or hidden representation of the nodes with the position encoding. Okay, and before we aggregate the node features into an edge feature, we add this position embedding to it, and then so we have a F-pooling layer to aggregate the neighbors of the edge into a edge feature, and then we also add a position embedding when we read out using MLPs to read out the final edge feature from it. And once we get those edge features, we can use the softmax function to construct a conditional distribution over these edges, and that is our conditional distribution construction. And that will finish this AR tree approach. And this is an overview of our AR tree. We start from the initial tree, right? And then we using this GNNs to aggregate those edge features, and then we use a softmax to get this conditional distribution, and we sample from this conditional distribution to get the edge to attach the next taxa, and we can keep on doing this until we arrive at the final stage that has all this text on the tree. Okay, and here's one example. The first one is on the tree probability estimation. So we did it on DS1, which is the standard data set. And we can see that the left and the middle plot shows the results from SBN and AR tree for the probability estimation we get from those two different methods. And compared to SBN, we see that AR tree can provide more accurate probability estimates for trees with high probabilities, okay, in the high probability zone. And also the performance tends to be more steady because it does not have these outliers like SBNs. That's because AR tree has less constraints in the modeling space. And we also tried to investigate the number of sample size for the performance of the algorithms. So you can see from the right plot, when the size of data sets increases, AR tree can provide more accurate estimation. But when the SBN baseline tends to level off, that means their model complexity is quite limited, and they cannot digest the information from more data sets. And we also apply this method across 8 benchmark data sets. You can see that overall, this DS1 to 8 data sets, AR tree can outperform this SBN baselines by a large margin. The next example is on Larson Bayesian phylogenetic inference. You can see from also the left plots, compared to SBNs, AR tree can provide better elbow and more accurate tree topology posterior estimations. And we did an ablation study about the order of the taxa, because for a sequential model this actually matters. But we find out that the order does not matter too much. Okay, so for all those kind of random orders, AR tree outperform SBN by a large margin. Interesting observation is that we said before, because every time you add a taxa, the node embedding is different. So we did an ablation study by turning off this time-guided readout procedure. We see that in this case, AR tree can be trapped in a local mode. So it does not realize the full potential of it. That means time readout procedure is crucial. And we also did a comparison to 8 benchmark data sets in terms of KL divergence, elbow, and the 10 sample elbow. This is the training objective, and also the marginal likelihood estimation. In novice criteria, AR tree outperforms SBN. So just to summarize, we have proposed a deep orthographic model for phylogenetic inference. That is completely standalone and has full support over this tree-to-white space and can provide better approximation than SBNs. Future work would be to improve the computation speed of AR2 because it's a sequential model, so it's kind of slow at this current stage, and to incorporate learnable topology features in the broader range of applications where efficiently leveraging the topology information could be of great importance. And that's it. Thank you.